be looking at sections 8.1 through 8.2. Our first one's on photosynthesis. This is specifically about the light reactions. So we can break photosynthesis up into two parts, like we did cellular respiration. Cellular respiration, we had glycolysis, the Krebs cycle, and electron transport chain. Photosynthesis will break up into the light reactions, the ones that require light, and the Krebs, or the Kelvin cycle. So, light reactions, Kelvin cycle. Kelvin cycle is kind of like the opposite of the Krebs cycle, building glucose from smaller organic molecules. The light reaction is going to get us the ATP energy and the electron carriers to be able to do the Kelvin cycle. And the photosynthesis happens into the chloroplast. And here's an actual picture of a chloroplast from a microscope, and here's an artistic rendition. So I would spend a little bit of time to familiarize yourself with these different pieces, components of the chloroplast, so that you can trace where everything is taking place as far as photosynthesis inside of the chloroplast. So what is photosynthesis? Plants don't eat, so they still need molecules, organic molecules, glucose, to get energy from, ATP, because they still do cellular respiration. But they create their own organic compounds from carbon dioxide and water. They use the energy that's stored in light in photons for this anabolic process, so building more complicated compounds, glucose, from simpler ones, carbon dioxide and water. So it's endergonic. It requires the input of energy from ATP and sunlight. So basically, photosynthesis on a molecular level is the opposite of cellular respiration. Carbon dioxide, or carbon compounds, are reduced instead of oxidized. They gain electrons. So here we have photosynthesis, and here we have respiration. In photosynthesis, carbon atoms are gaining electrons, gaining energy, input of energy to form C6H12O6 glucose. Water is oxidized. We're taking electrons away from water to give to carbon dioxide to form glucose. That's the opposite of cellular respiration. We learned about NADH and FADH2. We're removing electrons, oxidizing glucose, and splitting it into simpler compounds, CO2, using those electrons to form ATP energy. And finally, at the end of the electron transport chain, our oxygen is taking those electrons. So, chemically, it's the very opposite. Where do we get the electrons here to reduce carbon dioxide into glucose? Well, we get those from water. So one of the first steps in photosynthesis is we split water to oxygen, to hydrogen ions, and we use those electrons to reduce carbon dioxide and form glucose. Alright. So we talked about photosynthesis in the two stages. It's done in the light reactions and in the Kelvin cycle. So light energy enters a plant in the light reactions. In the light reactions, we make ATP and we store electrons in the form of NADPH, a close relative of NADH. So light reaction is kind of similar to glycolysis in that we make a little ATP, but we also store energy in NADPH to be used when we're going to make glucose in the Kelvin cycle. Light reactions convert sun energy into chemical energy by splitting water to harvest the H+ and electrons. And the electrons, remember, just like in cellular respiration, those are our energy, are stored in NADPH. That's like the plant equivalent of NADH. ATP is also generated by the light reactions because we're going to need ATP to input energy into the Kelvin cycle to form glucose sugar. Alright, here we go. So, something important about light. How do we get energy just from light? Light energy. So when a photon strikes an electron, the electron moves up to a higher energy level. Kind of a review of chemistry, emission spectra and all that. When the electron falls back down up at this high energy state, it's not very stable. Over time it will naturally fall back down to a lower energy state. It releases that energy that it gave up by falling back down in the forms of light. And that's going to be important. Because plants, the cells or the molecules in plants will absorb this light energy in the form of excited electrons. So, light excites electrons. Different colors absorb different frequencies of light. So electrons in those colors are bumped up or energized by different colors of light. So depending on the molecule, it takes a certain frequency of light to energize their electrons. Plants, this is all important because plants use many different pigment molecules like chlorophyll A, B, and carotenoids to absorb as many different frequencies of light from the sun as possible, so that's more potential energy, more energy that they can get from that sunlight and excite electrons. Because excited electrons is how the plant gains energy from sunlight. For example, here in a plant, the emission spectra of different, or the absorption spectra of different plant molecules, two types of chlorophyll, chlorophyll A and B, and then carotenoids absorb this amount of, or absorb energy most from this frequency of light. So light that you shine on a plant in this range would not allow photosynthesis to happen very well. So, plants absorb light energy in photosystems. Right, in photosystems. So inside chloroplasts, there are two complexes that absorb light energy. And those complexes are stored or are found in the membrane of the thylakoid. So each of these little discs are thylakoids. This is where photosynthesis happens. These photosystems are in this membrane of the thylakoid that absorb light energy and transfer that energy in the form of electrons into NADPH and ATP. There are two photosystems, photosystem 2 and photosystem 1. The first one we'll talk about is photosystem 2. And we're talking about 2 first because it was discovered, well, it's the first one on the chain, but it was discovered second. So photosystem 2 is the first in the chain. So photosystem 2 directly captures photons from sunlight. It contains many light-capturing pigments in its protein complex. So here is photosystem 1, a protein complex, inside the thylakoid membrane. And each of these green dots are different light-capturing pigments. So, different pigments that absorb different frequencies of light, and their electrons get excited and move to a higher energy state. When different light frequencies strike these pigments, it causes the electrons to jump up to a higher energy state and then fall back down. But when they fall back down, they release light and heat. And that in turn, that light and heat will excite another electron. That'll jump up and then fall down and excite another electron. That'll jump up on energy level, fall back down. excite another electron in a different pigment. So a kind of chain reaction, domino effect of excitation and falling back down of electrons will cause this energy to jump around this photosystem until it reaches the middle of this photosystem. In the middle, there are two chlorophyll A molecules that are the end of this domino of electrons. They receive this domino of electrons from all of the other pigments and pass those electrons on to this primary electron acceptor. All right? Chlorophyll A is called P680 because that's the frequency of light that it absorbs best. So the electrons that it receives and passes on to the primary electron acceptor are replenished by splitting water. So water that the plant uptakes is split into H plus and OH, or O. And the electrons from this O are transferred to this photosystem to replenish its electrons as they're passed on to the primary electron acceptor. How can that happen? Well, chlorophyll A is the most or best oxidizing agent in biology. So it can even take electrons from extremely electronegative element oxygen. It passes those electrons to the primary electron acceptor, and then the primary electron acceptor will move on and give them to the electron transport chain, ETC in plants. So the high energy electrons captured by the primary electron acceptor goes through this electron transport chain that's just like the one in cellular respiration. The electrons flow down a cascade of protein complexes, each more electronegative than the last, and give up some of their energy. In the middle, just like in the complexes in the cellular respiration electron transport chain, there is a cytochrome complex. This cytochrome complex pumps hydrogen ions against their concentration gradient into the inside of the cytoplasm. That does the same thing as like the protein complexes in cellular respiration. We're setting up an H plus concentration on the inside of the cell to form ATP by chemiosmosis. So in the cell membrane, it's not shown here, there's another ATPase molecule where these H pluses will flow back through and create ATP that will be used in the Calvin cycle. As these electrons pass down the electron transport chain, they power cytochrome complex to pump H pluses to the inside of the thylakoid membrane, and then these electrons are passed to photosystem 2. So those high concentration H plus ions on the inside of the thylakoid will then diffuse back through ATP synthase to the outside of the thylakoid and produce ATP. That will be used in the Calvin cycle. So then the electrons are going to go to photosystem 2, virtually the same thing. We'll go to photosystem 1, Virtually the same process as photosystem 2, the first one. Photons from sunlight will excite the electrons in their pigments. They'll jump back and forth until they reach a middle chlorophyll B acceptor that accepts 700 nanometer light the best. It has a primary electron acceptor. The electrons will flow into the primary electron acceptor. Except this time, the electrons from the primary electron acceptor will flow through or into a enzyme called NADP+ reductase, which will use those enzymes or use those electrons to reduce NAD+ to NADPH. So we're making NADPH, and we'll use NADPH in the Kelvin cycle. So here's the big idea. Light comes in at 2 points, photosystem 2 or photosystem 1. In photosystem 1, it jumps back and forth from these light-absorbing pigments until it reaches chlorophyll A. Chlorophyll A then gives these electrons to the primary electron acceptor and becomes reduced, grabs more electrons to replenish its supply from oxygen from splitting water. These electrons from photosystem 2's primary electron acceptor flows through the electron transport chain and through the cytochrome complex, pumping H+ ions from the outside of the thylakoid inside. That gradient then flows through ATP synthase, chemiosmosis, to produce ATP that we're going to use later in the Kelvin cycle. Meanwhile, those electrons continue their journey to photosystem 1, to chlorophyll B. Light that enters photosystem 1 will also jump back and forth from pigment to pigment until it gets to chlorophyll B, and then it will give those electrons to its primary electron acceptor, which will shuttle the electrons through NADP+ reductase, which will reduce NADP+ to NADPH, our stored energy, our electrons that we're going to need to build glucose in the Kelvin cycle. Alright, that's pretty much it. I know this is a little more complex than in cellular respiration, but we'll take a good long look at this in class as well. If you have any questions, just let me know.